My name is Denise. I am presenting from the Biomedical Engineering Department at Florida International University. The topic of my presentation is on the observation of calcific media combined with media from oscillatory flow conditioned valve endothelial cells leads to valve interstitial cell calcification. Our heart has 4 valves, each consisting of leaflets that facilitate unidirectional blood flow. The aortic valve is located between the left ventricle and the aorta and is the most commonly diseased of all. Calcific aortic valve disease is one of the most prevalent chronic heart problems that involves hardening of aortic valve leaflets due to calcium phosphate mineral deposition. This leads to valve stenosis, regurgitation, and overall reduced cardiac output. Valve endothelial cells and valve interstitial cells are major valvular cell types that reside on the outer and inner layers of leaflets. Hemodynamic environments on valve tissues can impact cellular interactions, such as communication between valve endothelial cells and valve interstitial cells. Flow oscillations are commonly observed on the fibrosa layer of the leaflet, while the ventricularis is mainly laminar. Studies have shown disturbed flow can result in pro-inflammatory responses and valve endothelial dysfunction, which can lead to phenotypic switching of quiescent interstitial cells to osteogenic phenotypes. This may be the reason why calcification generally initiates on the fibrosa layer. The two major components of hemodynamics are wall shear stress and oscillatory shear index. Past research has shown cell alignment differences between laminar and disturbed flow. Using flow oscillations as a key regulator, we would like to further investigate the degrees of oscillatory shear and its effect on valve endothelium and changes in interstitial phenotypes that can potentially lead to disease or remodeling. To determine the level of flow disturbance and its relation to calcification, we use the oscillatory shear index parameter, or OSI, to quantify changes in direction of wall shear stress. The OSI equation above shows a ratio between forward shear stress and total shear stress, and ranges from 0 to 0.5. This is an animated representation of OSI. At 0 OSI, fluid movement is either static or fluid moves steadily with no changes in direction, whereas at 0.5 OSI, fluid is in full oscillation. Any value in between can be considered moderate oscillation. We first obtained a vial of commercially available rat valve endothelial cells and expanded them in T75 flasks. The rat valve endothelial cells were then seated in bioflux plates that consist of 8 microfluidic channels per plate for 24 hours. Once cells attached, they were then conditioned for 48 hours under steady flow, 0.25 OSI and 0.5 OSI. Literature has shown shear stress magnitudes on the fibrosa layer to vary between 0 and 15 dynes per centimeter squared, and is dependent on the stroke volume as well as point and time of a cardiac cycle. A consistent average shear stress magnitude of 1 dyne per centimeter squared was applied to the endothelial cells for all flow groups. A separate static group was also conditioned for 48 hours in a regular culture flask. This is a side view of the Bioflux system. Cells are pumped into channels during seeding. The orange layer depicts the bottom of the plate, which is also the site for cell attachment. The input and output wells of the plate are connected to a pneumatic pump. By alternating pressures into these wells, fluid flow in the channels will change direction, therefore generating flow oscillation. To test the effect of paracrine regulation between valve endothelial cells and valve interstitial cells, we also obtained a vial of commercially available rat valve interstitial cells and expanded them in culture. The conditioned media from valve endothelial groups were collected and subsequently used to culture valve interstitial cells with equal volume of fresh media or pro-calcific media for 7 days, with one media change on day 4. The pro-calcific media used consisted of regular DMEM with 5% FBS and 1% pen strip, 1.8 millimolar of calcium chloride, 3.8 millimolar sodium dihydrogen phosphate, and 0.4 units of inorganic pyrophosphate. This media was combined with conditioned media from valvular endothelial cells in all flow groups to induce calcification in valve interstitial cells. Rat valve interstitial cell calcification was measured by alizarin red staining, as shown in image A. The alizarin red dye was then extracted and quantified with a microplate reader, shown in image B. An N equals 3 technical replicates were conducted for each flow group. Highest calcification was observed in 0.5 OSI group. Statistical assessments showed significantly increased calcification in the 0.5 OSI group compared to all other flow groups. Gene expression data from valve interstitial cells also show an upregulation of BMP2 and RUNX2 in the high OSI group. These expressions are associated with calcification responses. The static group also exhibited an increase in BMP2 expression, and this may be due to extremely low or zero shear stress, which is known to be associated with calcification in vascular systems. Previous studies have shown an upregulated alpha SMA gene and protein expression in valve interstitial cells at wound sites. This correlates with our findings where groups with high calcification expression also experience upregulation of alpha SMA. Our findings show that a combination of low shear stress at 1 dyne per centimeter squared with high OSI results in activated valve interstitial cell phenotype and substantial risk of valve calcification. We also see that valve endothelial cells exposed to low shear stress and low to moderate levels of flow oscillation maintain a quiescent valve interstitial cell phenotype via paracrine regulation. The results here conclude our two-dimensional cellular studies on OSI and valve calcification. Future work includes scaling up from two-dimensional studies to assessments of calcification in three-dimensional valve tissues exposed to various flow oscillations. I'd like to thank Hutchison Lab and Ramaswamy Lab at Florida International University. Thank you for your attention, and thank you BMES 2020. This is a mathematical and graphic representation of OSI. The OSI is equal to 0.5 times parentheses 1 minus absolute value of sum of wall shear stresses over the sum of wall shears. Gene expression data from rat valvular endothelial cells, valvular interstitial cells. Gene expression data from rat valvular endothelial cells. No!